The circulatory system is one of the most vital networks sustaining human life, and blood vessels serve as its intricate highways, carrying blood to every cell of the body. These vessels form a continuous closed circuit that begins and ends at the heart, ensuring the delivery of oxygen, nutrients, hormones, and immune cells, while simultaneously removing carbon dioxide and metabolic wastes. Blood vessels are classified into three main categories: arteries, capillaries, and veins, each uniquely designed for specific functions within this vascular system. Arteries are the vessels that carry blood away from the heart under high pressure. Their thick muscular and elastic walls allow them to expand and contract with each heartbeat, ensuring steady blood flow even between cardiac contractions. They are subdivided into elastic arteries, such as the aorta and pulmonary trunk, which absorb pressure surges through elastic recoil. Muscular arteries, such as the femoral and radial arteries, which distribute blood to individual organs. And arterioles, the smallest arterial branches, which regulate blood flow into capillary beds and are key controllers of systemic blood pressure through vasoconstriction and vasodilation. Capillaries, often described as the true exchange vessels, are microscopic and composed of only a single layer of endothelial cells and a basement membrane, allowing efficient exchange of gases, nutrients, and wastes between blood and tissues. Their structural variations, continuous, fenestrated, and sinusoidal, reflect their specialized functions, from maintaining a selective barrier in the brain to facilitating rapid filtration in the kidneys, or allowing passage of large molecules in the liver and spleen. Veins, on the other hand, return blood to the heart under low pressure. They possess thinner walls and larger lumens than arteries, and contain valves that prevent the backflow of blood, especially in the limbs where gravity opposes venous return. Venules collect blood from capillaries and gradually merge into larger veins that ultimately converge into the superior and inferior vena cava, delivering deoxygenated blood to the right atrium. Together, these vessels maintain a dynamic equilibrium in circulation. In laboratory study, examining and classifying these vessels helps students understand how each vessel type is structurally adapted to its function. Arteries for pressure resistance, capillaries for exchange, and veins for volume accommodation, revealing the beautiful efficiency of the human circulatory design. The microscopic anatomy of blood vessels demonstrates a remarkable structural adaptation that supports their physiological role in the circulatory system. Every blood vessel, regardless of size, consists of three primary layers known as tunics: the tunica intima, tunica media, and tunica externa, adventitia. The tunica intima, the innermost layer, lines the lumen with a thin sheet of endothelial cells that create a frictionless surface for smooth blood flow. This layer is vital for regulating vessel permeability, secreting substances like nitric oxide for vasodilation, and maintaining an antithrombotic surface to prevent clot formation. Beneath the endothelium lies the subendothelial connective tissue, and in arteries, an internal elastic lamina that provides elasticity and resilience. The middle layer, the tunica media, consists of concentric layers of smooth muscle fibers interwoven with elastic tissue. It is the thickest layer in arteries and allows them to maintain pressure and regulate lumen diameter through vasoconstriction and vasodilation, which are controlled by the sympathetic nervous system. Elastic arteries like the aorta contain multiple elastic lamellae to absorb pressure fluctuations during the cardiac cycle, while muscular arteries possess more smooth muscle to control blood distribution precisely. The outermost layer, the tunica externa or adventitia, is composed mainly of collagen fibers that provide structural support and prevent vessel overexpansion. In large vessels, this layer contains small arteries and veins called the vasa vasorum that nourish the outer wall, and nerve fibers, nervi vasorum, that regulate tone. Comparing different vessel types reveals key structural distinctions. Arteries have thick walls and narrow lumens to withstand high pressure. Veins have thinner walls, larger lumens, and valves to facilitate venous return. Capillaries, in contrast, are composed solely of endothelium and basement membrane for efficient exchange. Special vascular adaptations also exist, such as the hepatic portal system that directs blood from the digestive organs to the liver, and the arterial baroreceptors in the carotid sinus that sense blood pressure changes. In the anatomy laboratory, These differences are explored through gross dissection, vessel tracing, and histological observation. Under the microscope, arteries display thick tunica media with visible elastic laminae, while veins exhibit thinner walls that may appear collapsed. Capillary networks appear as delicate branching channels, demonstrating the close connection between microanatomy and function. This layered organization, intima for exchange, media for control, and externa for protection, illustrates how structure and function are perfectly integrated in the vascular system. The physiology of blood vessels centers on the dynamics of blood flow, resistance, and pressure, all of which ensure that every organ receives adequate perfusion according to its metabolic needs. Blood flow F is directly proportional to the pressure gradient, dP, between 2 points, and inversely proportional to the resistance, R, within the vessel, expressed by the equation F dP R. The heart generates the initial pressure that propels blood through the arteries, but the vessels themselves determine how that pressure is distributed and maintained. Blood pressure, measured in millimeters of mercury, mmHg, varies throughout the circulatory system. It is highest in the aorta and arteries, declines through the arterioles, and becomes minimal in the veins. The average arterial pressure, or mean arterial pressure, MAP, represents the effective pressure driving blood into tissues and is calculated as diastolic pressure plus 1/3 of the pulse pressure. Regulation of blood flow occurs through multiple mechanisms: local, neural, and hormonal. Locally, tissues can self-regulate their blood supply through autoregulation, where increased carbon dioxide, hydrogen ions, and lactic acid promote vasodilation to enhance oxygen delivery. Neural regulation is primarily mediated by the sympathetic nervous system through the vasomotor center in the medulla oblongata, which adjusts vessel diameter in response to baroreceptor and chemoreceptor feedback. Hormonal regulation involves several systems. Epinephrine and norepinephrine increase cardiac output and vascular tone. The renin-angiotensin-aldosterone system, RAS, raises blood volume and pressure. Antidiuretic hormone, ADH, enhances water retention and vasoconstriction. And atrial natriuretic peptide, ANP, promotes vasodilation and sodium excretion to lower blood pressure. Capillary exchange is another vital physiological process, occurring primarily by diffusion, filtration, and osmosis. Oxygen and nutrients diffuse from blood to tissues, while carbon dioxide and wastes move in the opposite direction. Hydrostatic pressure pushes fluid out of capillaries at the arterial end, while colloid osmotic pressure pulls it back in at the venous end, a balance described by Starling's forces. Venous return to the heart is facilitated by skeletal muscle contractions that squeeze veins, respiratory movements that create pressure gradients, and venous valves that prevent backflow. Disruptions in these mechanisms can lead to clinical conditions such as hypertension, hypotension, atherosclerosis, varicose veins, and aneurysms. Laboratory measurement of blood pressure and pulse provides essential insights into vascular physiology. Observing how blood pressure changes with posture, exercise, or stress helps students understand the dynamic nature of cardiovascular regulation and how the body maintains homeostasis despite constant internal and external changes. In the physiology laboratory, the study of blood vessels transforms theoretical knowledge into hands-on understanding through anatomical observation, physiological measurement, and experimental exploration. Students begin by examining gross specimens or models to identify major arteries such as the aorta, carotids, and femoral arteries, as well as major veins like the vena cava, jugulars, and saphenous veins. Microscopic slides stained with hematoxylin and eosin, or special elastic stains, reveal the three tunics and help distinguish between elastic arteries, muscular arteries, and veins. Observing these histological differences deepens understanding of structure-function relationships. A core lab exercise involves measuring blood pressure using a sphygmomanometer and stethoscope. Inflating the cuff around the brachial artery and slowly releasing pressure produces Korotkov sounds, which correspond to systolic and diastolic pressures. Recording these readings under different conditions, resting, standing, or post-exercise, demonstrates the adaptability of vascular regulation. Pulse examination, often done at the radial artery, assesses heart rate, rhythm and strength, providing clues about peripheral circulation and cardiac output. Capillary refill tests and Allen's tests evaluate peripheral perfusion and arterial integrity, while observing capillary flow under a microscope, such as in a frog's web or mesentery, vividly demonstrates microcirculatory dynamics and autoregulatory responses. Pathological correlations are essential in lab education. Atherosclerosis, characterized by lipid plaque buildup within arterial walls, can be demonstrated histologically. Aneurysms show weakened tunica media, leading to balloon-like dilation, and varicose veins result from valve failure, causing venous pooling. Understanding these diseases connects microscopic abnormalities to clinical symptoms. Students also learn about safety and ethical considerations, using sanitized instruments, handling specimens responsibly, and obtaining informed consent for human measurements. By the end of the lab, learners not only recognize anatomical structures, but also appreciate the physiological principles that sustain life through continuous circulation. They can interpret how changes in vessel structure or tone influence systemic blood pressure, tissue perfusion, and overall cardiovascular health. Thus, the laboratory serves as a bridge between anatomy, physiology, and clinical medicine, transforming blood vessels from static diagrams into living, dynamic systems whose integrity determines the health of every organ in the human body.